So, what we're going to be doing today is we're working our way up. And one of the things we saw in the introduction was that there's this organization to the body where we start out with really, really small things like building blocks, and then we put those together and it goes to the next level. We put those together and it goes to the next level, a bit like Legos. And so, chemical level is the lowest level, right? You talk about like the periodic table here and stuff like that, different elements, right? So, we have different elements that are the building blocks for what make up these things for molecules. And the molecules will make up things like, well, we'll talk about tonight, these things called organelles, which are the little units inside of cells. But the cell itself, what we're talking about today, is the smallest living unit in that whole organization. So obviously, different chemicals and elements exist, but they're not alive. Okay? Cells, each and every cell that makes a few of the different types that we're going to talk about are all individual living units. It's just with us, they all tend to work together to give us life, etc. So we're going to talk about cells. The next level up from that is tissues. So we have a bunch of cells that are usually attached to each other and working together for a particular function. That's called a tissue. And so there's like epithelial tissue, there's muscle tissue, nervous tissue, and connective tissue. When we have more than one tissue type together, then that's what we call an organ. So, for instance, what's a common organ you might think of? Yeah, liver, yeah, heart, brain, something like that. Skin is the largest external organ that you hear about. That counts because it's just more than one tissue type working together. Even a blood vessel, like an artery or something like that, counts as an organ because it's got epithelial tissue lining the inside of it. It's got smooth muscle which maintains the diameter of it. It's got connective tissue holding those other tissues together, etc. Okay? So, that's an organ. Then from organs, we have what are called organ systems. So organ systems is where we have more than one organ working together. So if we were talking about liver is actually like part of the digestive system. So what are we going to get some other digestive organs that you guys think of? Yeah, stomach, stuff like that, right? So all of those are different organs that work together for a particular function to help break down the food you're taking in and get those nutrients into the blood so that your cells can get those nutrients. So that's the organ system, and oftentimes what we're going to be focusing on in this class is at that level, organ system level. When you look through the chapters in the book, most of them are like, okay, skeletal system, endocrine system, cardiovascular system, etc. But each time we get to a new system, oftentimes in order to understand how that system does its job and maintain homeostasis, that stable internal condition, we have to understand how each organ helps out like a team. And how each organ is able to do its function. Each organ is made up of tissues and cells, and so it's really down to the cellular level that we can understand it. So for instance, with the muscles, right? We know that when you stimulate a muscle to contract, it shortens. It's very simple. When you stimulate it to contract, when you stop stimulating it, it relaxes. But how does an individual muscle actually contract? You have to look at the individual cell, and we'll be able to talk about how we have these filaments that slide past one another. So it's actually even down to the molecular level as well. So that's why we do have a chapter where we're just talking about cells. Now, when we get to these systems, we'll talk about cells that are all kind of different, right? Neurons are like these weird star-shaped cells. Skeletal muscle cells are really, really long cells, as well as cardiac muscle cells, which are branching cells that kind of connect to each other. All the cells, in a way, do a good demonstration of this whole idea of what we call form and function, basically anatomy and physiology. So whatever structure we have, it allows that cell or that bone or whatever to do a particular thing. So for instance, you can even think about like the different teeth that you have, right? So the ones that you have towards the front, on the inside of the canine, they're a little bit sharper and they're there for biting pieces off of things, like tearing things off of a big something that we're eating, right? And then you get to the back, and you have what are called malariform teeth. They're more flat, and those are there for grinding a big flat surface so that you can really mash things together. So they have different functions that are allowed by the different shapes or the different forms that they have. So we'll see that in cells, we'll see that in organs, etc. So when we get to the organ systems, we'll talk about specialized cells that have these specialized functions. But right now what we're doing is we're just kind of setting the stage instead. So we're going to talk about what a cell is generically and how it survives as a living thing, etc. And then later on, when we get to these other systems, they'll say, okay, you know about a regular generic cell, but this is an epithelial cell. You look how weird it is and how it allows it to do this particular function for a muscle cell, etc. Okay, so let us do that which we promised and look at the cell. All right, so the drawing that you see right here is the artist's conception of a particular cell. Now, if you were going to draw a cell, what you might do is you might draw a circle like this, okay? And then inside of it, you might put a little dot like that. So what's the dot represent based on your high school biology or anything like that? Right, so that's the nucleus, right? So the nucleus is going to have the DNA inside of it, which is going to be basically the blueprint for the cell, how the cell should be able to make particular things or how it makes other cells like itself, right? And then the edge of the circle that you see out here, you know what that's going to be? Does everyone know what that is? Online people. So let's see. Juan and Jasmine and Harold are around. You can ask like Anastasia or something like that. And Sasha. She's going to have to correct me about the pronunciation of her name. Jasmine typing. We'll see what she thinks that purple line is. Maybe Nina or Naomi might know as well. You can't see what I'm pointing at. The circle that I drew on the screen, do you guys see that? Right? I did a black dot and then I did a circle on the street. Yeah. So, that little circle on the outside, what do you think that is? One sees it. That's good. That's the first step. We all see it. Yeah, in essence, it's the border of what's going to be inside the cell and what's going to be outside of the cell. Now, if we were a plant cell, we call it a cell wall. In an animal cell like ours, we just saw it like that instead of cell membrane. Okay? But it's kind of like our skin, right? Our skin is the border between what's outside of us and what's inside of us. And it helps to regulate what can get inside and what we want to keep out. Okay? So the cell membrane is a bit like that for the cell. So it's separated. What we see there is ECF. It's called the extracellular fluid. So extra, you guys know how to spell that, right? Cellular, that's actually one word, fluid. Like that. That's ECF. ICF is intracellular fluid. Intra, I-N-T-R-A, means inside, like interior. Extra means outside, like exterior. Okay? So there's fluid inside the cell that we see here, which is called cytosol. This is the intracellular fluid, that orange that we see in there. And then there's the fluid that's going to be outside that we see kind of in white. That's going to be extracellular fluid. So to separate intracellular from extracellular environment, and like we said, like our skin, it controls the passage of things in between. Okay? Okay, so that's the first kind of important component of the cell, is that which creates the border there. When we look on the inside of the cell, it's a bit like us. We say that we're a living thing, and we've got these organs that help to keep us alive, etc. But also, a cell is a living thing. So it has these little mini organs that we call organelles that are inside of it that each have a particular job. Okay? So, when we look inside the cell, what we have is this gel-like matrix, which is called cytosol, that you see underlined up there. And then the cytosol plus all these little mini organs or organelles equals this thing that we call cytoplasm. And any place where you see cyto, C-Y-T-O, that means cell. Okay? So, what you see listed there with all those bullet points are different organelles or mini organs that occur inside the cells that keep them alive in some way. So we're going to be talking about what each one of those things do. So it's a bit like we said, how we'll talk about our own organ systems and organs. But before we do that, we need to talk a little bit more about that plasma membrane, because the plasma membrane that you see there, when I drew it before, it looked very simple. It was just purple lines. But now, when it was this figure here, it looks a whole lot more complex, okay? So the orange that you see down below, that's the intracellular environment, okay? So down here, you see that intracellular fluid. Up here is the extracellular fluid, right? You see extracellular and intracellular. So that's going to be, the cell membrane is going to be the border between them. And you can see it's made up of a lot of different molecules. Last time we talked about weed, that's the royal weed, talked about the different molecules that you may have, things like carbohydrates and lipids and proteins and things like that. Okay? And the plasma membrane is often, if we're talking about those different types of molecules, this is actually made mostly of lipids. Okay? And so lipids are going to be things like phospholipids and fats and cholesterol and things like that. So when we look at this, you can see that there's these two layers of kind of these blue balls there with these two little almost strings coming out of them. Those things right there are called phospholipids. Okay? So those phospholipid molecules, they have, right, a circle. This is called a polar head to it. It's got a polar head to it. And then, in addition to that, it's got these little fatty acid tails to it as well. So, have these little tails extending out of them. So that's a phospholipid molecule that you see right there. And you can see how they're arranged in two different layers. One here and one there, and they have their tails pointing to the inside. Okay? So, most of the membrane itself is made up of these phospholipid molecules, and oftentimes what we call this cell membrane, we say that it forms a lipid bilayer. Bi like two, right? Bi about two wheels. A bilayer has two layers to it. One there, where the blue balls are, and one down below. So it naturally forms a lipid bilayer like that. And towards the outside and the inside, those blue heads they're basically water-loving, right? They're called, I'm going to type it in instead, so you don't have to look at my handwriting right now. But they're basically called hydrophilic. So hydro means water, philic, like phile means to love, right? So bibliophile, actually the person who loves books and things like that. So the heads are hydrophilic, and that's good because the fluid inside and outside the cell is mostly made up of water. So we want that part of the molecule to get along with water because it's next to water all the time. But the inside, where the tails are in the middle here, those tails are hydrophobic. And so if you hear somebody that has a phobia, right, like agoraphobia, they're afraid of going into open spaces, or arachnophobia, they're afraid of spiders or something like that. Phobia means afraid. Hydro means water. So hydrophobic on the inside means that it's water-fearing. And what that means is that inside is a bit like an Oreo cookie, right? You got the fat on the inside, so you can see wafers like that, right? And because it has that hydrophobic core to it in the inside, molecules that are dissolved, floating around in the extracellular fluid or the intracellular fluid, can't pass through there. They don't get along with that hydrophobic core. And so that has to have a good regulator or control as to what can go into and out of the cell. Not everything can just slip in and out. Because the hydrophobic core is going to make sure that we can control what goes in and out. So in a way, right, this is the border itself. Much like, if you want to consider it, like the border like between the U.S. and Mexico. Let's see. So, realize that the border that we have between the U.S. and Mexico is meant to not keep everybody in and out, right? So you can go to Mexico. Have you ever been to Mexico? Right? I was down in Puerto Penasco doing the study abroad marine biology program just about a week ago. Okay? You can go across that border, but we have specific areas where you can do so, and it's controlled. They want to see who's going in and who's going out. You control that. Same thing there. Okay? We want things to go in and out of the cell. The cell needs nutrients because we need to get rid of waste products, etc. But we want to control it. So the access points are usually these things here. So you see that orange thing right there, this channel? Okay? Those things, like all these red and orange things that you see around here, these are proteins. So in addition to lipids, phospholipids, we do have a lot of proteins that are embedded in the plasma membrane. And oftentimes where those proteins exist, it's like a little tunnel or a gate that will allow certain molecules to come across, go in or go out. So they're very important as, kind of checkpoints to let things in and out of the cell. Okay? So we see mostly phospholipids. We see some proteins that are embedded within it. We see even some other kinds of lipids or carbohydrates attached to it. Oftentimes they are used for identification for the cell, so that we can tell that the cell belongs in our bodies and it's not a foreign thing, things like that. Okay? But you can see there's a lot of different molecules that make up this plasma membrane. It's a lot more complex than we originally thought. Okay. So what we're going to do now is move into talking about some of these different organelles, the many organs that are inside the cell that help to keep it alive. We're going to talk about what they do. We'll wrap up at the end talking about the nucleus, because the nucleus is pretty complex. Okay, so we're not ignoring the nucleus, we're just going to do it at the end. So the first one that we talk about in terms of the organelles is called the rough endoplasmic reticulum. And so sometimes, as you can imagine, that might be abbreviated ER, endoplasmic reticulum. And you should probably spell it out so that I know that you know that it's endoplasmic reticulum as opposed to emergency room. Okay? But there's two types of endoplasmic reticulum. There's rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum is called that because it's a series of membranes like this attached to the nucleus. And then the rough endoplasmic reticulum, it has all these things that are called ribosomes, where you see the underlying word, attached to it. So if you look at it in the microscope, like you see in the upper right-hand corner there, it looks like it has all these little bumps to it. It looks rough. Okay? So that's where the name comes from. And what it does is it's a series of membranes that will produce protein. Now technically, and I actually heard about this at one talk that I was at over the last couple of days, we're going to see that there's rough endoplasmic reticulum and smooth endoplasmic reticulum. Now, rough endoplasmic reticulum, you can see here, the ribosomes are the main site for protein synthesis. So in order to take those amino acids we talked about yesterday and link them together to make protein, that's where that happens, right, in those ribosomes. Okay? So it's a big place where we make proteins. These are proteins that could go in the cell membrane, right? As those wear out, we've got to put more. It could be proteins that this cell is going to put out, by maybe their hormones or something like that, and we need to put them out. Or digested enzymes, that will digest fast or something. Those cells, like in the stomach, are going to put out proteins out of the cell. So they're made right there, the protein factories. So that's what the rough endoplasmic reticulum is, protein factories. Now, smooth endoplasmic reticulum is also membranous like that. It's attached to the rough endoplasmic reticulum, but it doesn't have ribosomes attached to it. Okay? So, it's not going to make protein, because we said the ribosomes are the protein factories. That's where we're actually cranking them down. The smooth endoplasmic reticulum oftentimes is going to do more what we call lipid synthesis instead, right? Lipids are fat. And what this does is it packages new proteins in transport vesicles. A vesicle is basically a bag. So like if you go to the grocery store or something like that, or maybe a cart, we consider it that way. When you go to the grocery store and you buy a bunch of stuff, you don't want to carry them all individually. You're going to put them in a bag. So when the rough endoplasmic reticulum is cranking out a bunch of proteins, we don't want them floating all over the place. We want to put them in a bag. Okay? And that bag is made, much like the plasma membrane or the cell membrane, is made up of like little phospholipids that encircles the protein and keeps them all in one place. So the smooth endoplasmic reticulum manufactures things like lipids, and it makes these different what we call transport vesicles, where we can take all these proteins that we've made and we can move them as a unit within the cell. Okay? Now, the thing, the kind of update that I was working about in the last couple of days is oftentimes we separate these there. We have rough endoplasmic reticulum, protein synthesis. And then for our smooth endoplasmic reticulum, it's lipid synthesis instead. Technically, the rough endoplasmic reticulum does protein and lipid synthesis. Smooth endoplasmic reticulum just does lipids, but it doesn't have a ribosome. So usually, in the past, we were talking about them separately. Protein there, lipid there. Technically, the recent update says that rough endoplasmic reticulum does both protein and lipid synthesis, and smooth only does lipids. But those are the endoplasmic reticula, rough and smooth. Alright, the next organelle we have seen here in orange is called the Golgi complex. The Golgi complex is also involved with things like proteins, okay? So one of the things we're looking at is that if we're making proteins in the cell, what we usually do is we have a nucleus that is kind of the brains of the cell. And the nucleus has the recipes, if you will, or the blueprints on how to make protein. Okay? So if you were talking yesterday, in order to make a protein, we have these 20 amino acids. It's like going to a feed store, right? So you go to a feed store and you have all these little pockets that are little, drawers of beads. And if you have 20 beads, you can take different beads and you can put them together. And the same 20 beads, but you put them together in different combinations, right? You get different necklaces. Could be, here's 20 of one, and it looks nice, it's all white, or maybe you do alternating. I'm just doing these three and alternate them or something like that. Proteins are the same. You only have these 20 amino acids, but it makes a big difference as to which one and what order as to what the protein is. And those instructions are in the nucleus, okay? So the nucleus has the recipe for how to make the protein. Rough endoplasmic reticulum is the factory for doing it. Smooth endoplasmic reticulum takes those lipids and surrounds it, makes what's called a transport vesicle, so you can carry it like a cart, if you will, in the grocery store. The Golgi complex here is where we actually package it again, but this time we're packaging it into what we call secretory vesicles. So secretory vesicles are like when you have a check pan and they put them in the bags for you to take them out of the store. Okay? So these are proteins that we made back here in rough endoplasmic reticulum, but maybe, like I said, these are digestive enzymes that we're not using the enzymes to sell. We need to secrete them into the stomach so that they can digest protein. This is the way that we can get them to the cell membrane and then dump them out outside of the cell. Using the Golgi complex, we kind of do a finishing process, we kind of clean up these different proteins to make sure that they're all ready to go, and then we package them using these secretory vesicles that will then be transferred to the plasma membrane. And then they basically open up and dump out their proteins into the extracellular environment. Okay? So it's a bit, like I said, it's a bit like a factory, right? So you've got, the designers that are in some offices, okay, this is what we're going to make now. We're going to make iPads or something like that. This is how we're going to do it, right? Rough endoplasmic reticulum is like a factory where they're putting it together. But once they make it, they have to put it in a package. That's a fit, if you will, like getting it in the package and finishing it up, and then maybe putting it in a box for shipping. And then, for that, we've got to take it over to the shipping department. We put them in big crates or something like that on pallets and stuff. And we take that to the truck, and then the truck basically takes it out of the factory. That's kind of like the Golgi complex, right? Packaging it together in big packages and taking it out of the cell. Okay? So all three of these things, organelles we've been talking about so far, are involved in some way in protein production and manufacturing, etc. So it's a pretty important job in the cell. Okay, so that's three organelles so far. Rough endoplasmic, smooth endoplasmic reticulum, and then Golgi complex. Fourth one we have here is a mitochondrion, and a mitochondrion, or mitochondria, if I'm plural, they are, this is a little phrase that commonly goes with it, called powerhouse of the cell. Okay? So, they're the energy organelles, and what they do is they are the major site of ATP production. So you guys remember that at all? ATP? A little bit. Mm-hmm. So ATP stands for, if I can type right, adenosine triphosphate. Okay? That's ATP. So what those organelles do, they're kind of bean shaped. You can see that they have this double membrane on the inside of them, goes back and forth and back and forth. And they produce this ATP molecule. And an ATP molecule is a bit like a rechargeable battery, like in your phone or your device or whatever. And so what you do is you normally take your phone and at the end of the day you plug it in, right? You plug it in and you take energy from the wall and you store it inside the battery of the phone. So that way the next day, when you need energy later on, you've got it to make calls whatever you want to do, right? basically how much fun you're having in your 560 class, right? Alright, so in a way, that's what's going on in mitochondria as well. It uses energy like from food you eat and things like that, in order to synthesize this ATP molecule. So it takes some energy to make ATP, and the tri part means three, right? There are three phosphates that are attached to this molecule. In order to make an ATP molecule, what we do is we take an ADP molecule, diphosphate, it has two, take another phosphate, take some energy, and glue it on there, okay? That energy that it took to kind of glue it on there is now stored in the ATP molecule, just like your battery in your phone. So now, once we have an ATP molecule, we've got, just like your battery, you can take that molecule, just like you take that battery, and you can use that energy at some other time or some other place in order to fuel what the cell has to do. Okay? So that's what it does. It cranks out ATP all the time. And then the cell will use that ATP in order to fuel other things that it has to do. Okay? So that's its big job, is manufacturing ATP. Alright. Now we get to a couple other molecules that are quite important, but they're a little bit simpler in terms of their structure. Rather than being big membranes all over the place or these bean-shaped things, these are just little bags. One's called a lysosome and one's called a peroxisome. And these are little bags, which means they have phospholipid molecules around these quarters of them, like we talked about before. And the lysosomes are basically like an intracellular digestive system. So what is intra meaning in? Interior, right? Inside. So this is a digestive system that's inside the cell, right? It's just like us, right? You have digestive organs, etc. Here's a digestive organ. So lysosomes have enzymes inside of them that break down substances in cellular debris. Okay? So if there's parts of the cell that wear out, or like the cell that manufactures things that's got different kind of remnants that need to get rid of, these lysosomes are bagged with enzymes, and they basically take those parts and put them into the lysosome, and it kind of breaks them down, just like our stomach, if you will, breaks down different things for us. Okay? So these are lysosomes, intracellular digestive system. We also have peroxisomes, and peroxisomes are kind of similar in the bag, but these ones are actually using a different set of enzymes that detoxify waste products. So for waste products, right, we have, for instance, like a urinary system. It works if we have different waste molecules that are in our blood and things like that, and kidneys take them out, and they try to make sure that they don't accumulate too much. So the peroxisomes work in a way kind of like our kidneys, and helping to get rid of waste products inside of the cell. And again, you can see both of them look like little bags, right? But inside those bags are special enzymes that work to almost like, not acids, but different types of clean chemicals that help us do chemical reactions and break these things down. Okay? So lysosomes and peroxisomes, little bags that inside the cell that help the cell survive. So there's these different organelles, mini organs, that help the cell do various things. But when we're comparing it to our body as a living thing, we also know that we need a lot of structure inside of our body. And so for us, we have a skeletal system, and then we have a muscular system that works with the skeletal system to help us move. The muscles that we have, skeletal muscles, all attached to bones, and when they contract, they're going to move the bones. Okay? The cell also has a skeletal system. That's weird. So cyto, remember, means cell, and then there's the skeletal, the support system. So the cytoskeleton inside the cell is not made up of bone. We'll talk about skeletal system and bone tissue later on. But the cytoskeleton inside the cell is made up of protein. So remember, all those other organelles that are making protein, a lot of proteins actually go to maintain the cytoskeleton of the cell. So that, a neuron actually retains its star-like shape, so it can communicate with a lot of other cells. So a muscle cell can actually retain its shape and move things inside of it, etc. Okay? So the cytoskeleton is a protein network inside of the cell. And there's different types of molecules that are part of the skeleton. So you can see, for instance, there are what are called microtubules, and you can see how there's an opening inside, right? There are microfilaments one's going to have different functions as well. So for instance, microtubules, like I said, form these little tubes, and in some cells, they do various things, like for instance, transporting secretory vesicles between cell regions or transport vesicles, right? So like the smooth end of plastic reticulum making back to protein, it doesn't just like scroll them out in the cell and they're bouncing around in there, right? There's actually almost like little lanes of proteins, like little rails, like a train system that runs through the cell that these vesicles will attach to, and they get moved along the rail, and they go from the smooth end of plastic reticulum to the Golgi complex, and from the Golgi complex straight to the cell membrane. And so it kind of encapsulated like that when you move. So that's one of the different functions of, for instance, microtubules. We'll find out later on that a lot of cells have these little projections coming from them that are called cilia or flagella. Sperm cells that we'll learn about at the very end have one flagellum, right? Like a tail, right? That tail itself is made up of microtubules. And cilia, when we have a cell in cilia, it doesn't have one long one, it has a bunch of little ones, but they move on the surface of the cell. Whereas the flagellum will move a sperm cell along, it propels the cell. Cilia will actually move things past the cell, almost like, like crowd surfing, right? So when you get that, I can get through people's arms, it's moving you along. Same thing, so if you have a cell with all these cilia, they can move particles or mucus or something like that past the cell as part of that tissue, okay? And those things are examples of different types of microtubules. Microfilaments are also part of the cytoskeleton. They generally have a bit more to do with moving things. So for instance, when we talk about muscle cells, when you have muscle cells, it's all these little filaments inside the muscle cells that will slide past one another in order to allow muscle cells to shorten. Those are made up of microfilaments. Okay? So they're a bit smaller, a bit thinner compared to our microtubules. And then, the last one we saw there was the intermediate filaments. And their name comes from the fact that they're in between in terms of their size, right? They're bigger than the microfilament, smaller than microtubules. And the best example we have from an intermediate filament is keratin. And keratin is a protein that we find in our hair and in our fingernails that makes them stronger. And as we said, like the cytoskeleton is there for protection, strength, structure, and then also sometimes for movement. Okay? So those are three different examples of our cytoskeleton, proteins that make up our cytoskeleton. Right? Okay. So, questions about organelles that we've seen so far? Okay? So you need to know, be able to recognize those different types of organelles and be able to describe, at least in a sentence or so, what they do. Okay? Okay. So, our last one we were talking about is going to be the nucleus. Now, the nucleus, we said, is kind of the brains of the cell, and it is an area where we have this big recipe book, okay? And that big recipe book is basically the DNA. And like a recipe book or a cookbook, right? It's got all these different recipes inside it. So if you want to know how to make, right, fettuccine alfredo, you're going to go to page 113. If you want to know, like spaghetti bolognese, you're going to go to page 127. Right? And so all of those are in this big cookbook. And the same thing with the DNA. The DNA is a series of these nucleotides that are all sewn together, but it's like a cookbook. So you go, it's broken into these different functional sections called genes. Okay? And so this segment is one gene, and that's the recipe on how to make this particular protein, etc., or how to do this particular function. Okay? So, that's in essence kind of what the DNA is there for. It's got all these genes in it that allow the cell to use a particular function, the recipes for those things. So, it's got two major functions in it. Number one, it guides the synthesis of various proteins that have all these recipes on how to make them, whether they're for microtubules or for a channel in the membrane or something like that. And it's also a blueprint or a recipe for how to make a new cell. So, when we have a muscle cell, when we, have, for instance, or maybe a skin, say you guys have your skin, When that happens, what we want to do or heal is all those cells that were damaged or removed, we want to replace so that your skin looks the same again and has the same function. And so, those cells that are near the area where you got cut, they have to rapidly divide and make new cells. Well, we want those epithelial cells to make more skin to make more epithelial cells. We don't want to start like layering on neurons there or muscle cells on the surface of our skin or anything. We want the same ones that we had before. So DNA also allows us to take the original cell and then have the blueprint on how to make an exact copy of the cell. Okay? So that's the major function. Number one, you have recipes for different types of proteins, etc. Number two, you have recipes for how to make a new cell. Okay. So, that is where we find our DNA, which is that big cookbook. And do we know what DNA stands for? Online people? Let's think about it. They're Googling it right now. You see how fast they get typed. Need to get help them. It's a long word, so just talk back and just give it a second. Starting with D. Yes, exactly. So you got to type that very quickly. That's very good. So deoxyribonucleic acid. Okay. So D is the D part. Okay. Deoxyribo, right? Just talking about different components that make it up. And then nucleic at the end, and then A is acid. Okay. So deoxyribonucleic acid. If you have chromosomes, you like that, if you look like X's and things like that, sometimes see, that's made up of DNA. Okay. So that's the basic cookbook. But also, we find that there's another type of nucleic acid that is called RNA. You see here, okay? And RNA is a bit like DNA. DNA, you guys all see that picture of like a twisted ladder, you know, like the double helix thing when you talk about DNA? Okay, so it looks like a ladder that twists around and stuff like that, okay? RNA, rather than being a double helix, is just a single strand instead, okay? In RNA, there's three different types of RNA, and their role is to work with DNA to help make protein. Okay? So you can see there's three different types of RNA, and that's just ribonucleic acid. It's made slightly differently, and it only has a single strand, that's what you're talking about, rather than a double strand. So that's a slightly different name. But there's three different types of it. And the way that I tend to think about it is that with the DNA, as I said, like a cookbook, right? So say you wanted to, make crepes or something like that, and you had a friend who knew that there was this cookbook in France that had the recipe for the best crepes, right? So you don't use French, but there's this cookbook in France that has the best recipe, so you would like to get that recipe in order to be able to make those crepes. So, the DNA is like that cookbook. That cookbook is you yourself can't read. So what you might do is you might have somebody that lives in France go to the book, you call them up, you email them, and say, can you go read the recipe for the crepes and then just send it to me by email. Okay? So that person speaks French, no problem. So they go and find that particular section of the DNA, they read that gene, okay? And they say, okay, this is the recipe on how to make those crepes. Okay? They send it to you, but it's in French. So you have the recipe now, but you can't read it, you can't make it yet. So the mRNA that we get is called messenger RNA. Okay? Messenger RNA is like that friend you have in France that can read the cookbook and read the DNA, but it doesn't get you to where you need to be yet in order to make the crepes. Okay? So that's a messenger RNA, that's mRNA. So it does what's called DNA transcription. So the transcribe, if you're a transcriptionist, you're basically writing down what people are saying, like in court, right? So messenger RNA does that. It will go to the particular section of the DNA, read off that recipe, and then says, okay, here's your recipe. Okay? Second one is called ribosomal RNA. And so ribosomal sounds like what? Ribosomes, exactly right. Ribosomes are exactly the reference bottom ER, where we make protein. So those ribosomes, we say, those little bumps, those are basically made up of ribosomal RNA. And what they do is they read the recipe. So it's a bit like, say you get your email with a French recipe, but then you have a friend here that's Canadian. So what they can do is they can read French, but then they can translate it for you so that you understand what you're supposed to do. Okay? That's a bit like these ribosomal RNA. Okay? They're there, and they're there to read it out and say, okay, this is what I'm going to need. I'm going to need 2 cups of sugar and 3 cups of flour and some baking soda and this and that, right? Okay? So that's what's going on inside of those ribosomes. It's in essence, you've got now a recipe for all the different things you need in order to make that protein. But the last thing is the tRNA. The tRNA is called transfer RNA. Okay? And so if you're in there as a master chef and you finally have your recipe in the kitchen, what you might have is a friend of yours who's helping you make these things. So you're reading the recipe, you're like, okay, I need 2 eggs over there, I need the sugar from on the shelf over there, and that person is going to get those different components and bring them to you so that you can then make that particular recipe. Okay? That's the role of the transfer RNA, right? Inside the ribosome, where all that's happening, but the ribosome RNA is basically talking to the transfer RNA and saying, okay, go get me, this amino acid, go get me this amino acid, right? Go get me alanine, go get me thymine, and stuff like that. And then I'm going to put them together and start making this particular protein. Okay? Each of those RNA molecules each have a slightly different function. Messenger RNA is reading DNA, right? Ribosomal RNA is kind of like the kitchen, which is going to be the cook is going to be putting together. And then the transfer RNA is kind of like the sous chef or the assistant chef who's going to get those amino acids and bring them to the site of synthesis of the ribosome. Okay, so DNA and RNA. DNA is found in the nucleus, and RNA is found in the nucleus to begin with, right? Reading off the recipe, but then in the ribosome. So you see this picture here, we can see all the different types, right? There is the DNA, or twisted double helix thing. Here is the mRNA, the messenger RNA. So you see it's reading from here to here. This is the recipe right here. Okay? Then we can see our messenger RNA goes out of the nucleus, loses the nucleus. And then this, let me get the color. This ribosome right there, that's the ribosome. That's the ribosomal RNA. That's the kitchen where all this cooking is going to happen. And then these things here, those are the tRNA molecules. They're going out and grabbing amino acids and bringing them to the ribosome so we can start assembling that polypeptide chain that will become a protein. Okay? So DNA, messenger RNA, and then rRNA, ribosome, and then tRNA, all in that picture right there. And the recipe, you'll get part of this if you take like that general biology class, but the whole recipe that they're talking about in terms of the mRNA and stuff like that, it's actually in 3 nucleotide chunks. So these are called codons. And so what this is, we have this one, and the code, so like a piece of code where you have this number equal to that letter and stuff like that, because that's 3. So if you have AUG, that basically signals the beginning of the recipe. If you have GGC, then that is the code for liking that particular amino acid, etc. So we're reading along the mRNA, and that's telling us which of these different amino acids you see on the right are supposed to be part of this protein. Okay? So they're called codons. They form one of those codons. Okay. So balls are kind of interesting. You won't see them on the exam or anything, but the balls, one of the things about human anatomy physiology is on a certain level, when people look at it, they think, wow, everything's saying human anatomy for hundreds of years once we discover all the internal workings and stuff like that. What they're going to do is learn anymore. We already know. It's not like a new bone of the forearm or anything. But there are still some things that they're discovering, particularly like the nervous system. But even some stuff, there are things that they still don't understand. They found these things in the last 10 years. They look like a techno mineral. They call them gold. They know that they think it's stored in there, but they don't know exactly what. They know that they have to go huge amounts of the nucleus. They think maybe they might be used for transferring, for instance, the different parts of the RNA or something like that, but they're not sure. So they even microscope, they can see the different than ribosomes or anything else, but they're not exactly sure the function. So this goes to show you they're still very new and interesting things that they're discovering. Okay. So that being said, what we've done so far is we've got our picture of the cell. So we've got our cell membrane, we've got the different organelles inside and what they do. So now we have a basic understanding of what's going on with the cell. So from here, what we're going to do is we're going to talk about a little bit about how things go into and out of the cell, etc., how we make new cells, and things like that. But before we do that, we're going to do a break. Because it's been a while. Okay? So, right now, what we do is, according to that clock, 1:40. Okay? So, why don't we... So, why don't we take a 15-minute break? Okay? So, come back at 5 until the hour, and then we'll continue on. We'll finish up the chapter on cell, and we'll move into the chapter that is our first system of the body, which is the integumentary. Okay? Okay, so vacuums are out there, you can come outside, you know, warm it is, something like that. People at home can run outside if they want to. And we'll start again in 15 minutes.